I'm very excited to introduce our first guest, Andrei Roibu. I have to say, he's a PhD student, though you wouldn't believe it looking at his LinkedIn profile. He's currently working at Oxford, and his research is based on mainly deep learning and advanced analytical pipelines, particularly looking at brain aging and looking at the external factors on brain aging. And he's worked actually for several companies, a huge number of companies, Rolls-Royce, but also the European Space Agency. He's also worked at Siemens and knows well the Wellcome Trust, which is a major charity in the UK. And he also is in a STEM outreach program which helps people who would not be naturally exposed to science in Romania to actually work and get to know a bit the science. Today, he's going to be talking on brain ages derived from different MRI modalities and associated with distinct biological phenotypes. Thank you for that. But basically, if I can say, he's going to actually be looking at the fact that the brain is quite heterogeneous in its aging, both temporally and spatially. And then you have the fact that as your brain gets older, it might not match your chronological age. So this is called delta in terms of aging. And he's going to be looking at the external factors of this and how this affects the brain aging. So I'll stop talking about it and let Andrei talk about it. Welcome, Andrei.
Thank you for the introduction. You've already given half of the talk at that point. Also, as you noticed, I've changed the title because my fiance saw it and thought, mate, no one's going to come to your conference talk with that kind of title, so get something more catchy out there. But yeah, thank you very much for the introduction. So today, I'll hopefully take you on a journey in one of the most complex structures in the universe, which is the human brain. And in the story, as was mentioned, we'll be looking at brain aging, which, although is a phenomenon that impacts every one of us, is actually not very well understood at this point. So by using the magic of deep learning, advanced data analytics, and all that fancy stuff, hopefully by the end of this talk, I'll give you an understanding of why the age of your brain might not match the number of candles on your birthday cake. So first off, go on. No? Does it work? No? All right. Well, while this gets working, I'll start off by asking you a question. Try again? No? No? All right. I'll start off by asking you a question before we get there. So if you can put up your hands, do you think the age of your brain matches your chronological age? Or is it younger, is it older? So who thinks your brain age matches your age? Put your hand up. Okay, a few people in the audience. Okay, quite a lot, actually. Who thinks that your brain is older than your chronological age? Okay, one over there, a couple over there, okay. And who thinks they're younger? Yay, okay, quite a good group. Cool. So, obviously, our brains are aging as we do. However, as mentioned, this is not a linear process. It's actually quite heterogeneous. And we're seeing variations across populations. And this could lead to situations in which you see young people suffering from neurological conditions like dementia. But at the same time, you could see very old people, like my great-grandmother, 97, who's as healthy as she can be from this point of view, at least. But moreover, we see in literature that certain factors, such as substance abuse, exercise, diet, things such as the presence of neurological conditions or chronic conditions, genetics, and environmental factors, all of these have an impact on if brains appear to be younger or older. But at this point, you might be asking yourself, okay, but how do we actually see these things? And this is where different techniques such as MRI, non-invasive techniques, come into play. Now, in a nutshell, MRI works by using large and powerful magnets plus radio frequencies to get different images of the brain. And by tweaking the parameters of acquisition in an MRI machine, we can get different types of images sensitive to different information. And we refer to these types of images as modalities. Now, the most common modality is structural MRI, which, as it says on the box, looks at the structure of the brain. Things like white matter, gray matter, the stuff we've all learned in school about. But there's other stuff out there into the wild. So we have things like susceptibility weighted MRI, which looks at the deposition of certain elements in the brain, like iron or calcium. We have the fusion MRI, which looks at water diffusion through the brain and gives us information about the white matter, so things like structure, microstructure, and connectivity. And we have functional MRI, which by looking at blood oxygenation levels, gives us information about the activity in the brain. And this could be either at rest, and we get resting state, or during a task, and we get task, functional MRI. But this is not the full story just yet, because we can further process these modalities to get summary measures of interest. And we refer to these as contrasts. And there's a lot of contrasts out there in the wild. But just to give you an example of what we might be seeing here, for example, for the fusion MRI, we get things like tractography maps, which tells us about the connectivity. We get anisotropy maps, which tell us about the structure and microstructure of the brain. And we get skeletonized masks, which are population invariant, so they project the information on a skeleton. But why am I telling you all this? Well, it's because all of these different modalities and these different contrasts encode different types of information about the brain and possibly about brain aging. And they're relevant because when we look at the history of brain aging, what we see is that from about 140 studies in the past decade, about 75% of them only used information from structural MRI. But this is not the full story just yet. Because if we only look at those studies that used volumetric information, that's 2D or 3D volumes, that changes to actually 95%. 95% only used structural MRI for brain age prediction. So you might see the problem here, which is, for one, we're losing all that information that's captured by all the other modalities. But two, you can see there that's only 39 studies out of 146 that use volumetric information. So any information captured in the spatial context of those images might be lost by not using volumetric information. So that's where our work comes in. And what we're doing is essentially, what we want to do is use 57 different contrasts. So you remember from the previous slide the contrast. 57 different contrasts to try and understand brain aging. And these are derived from 5 core modalities. Now, for this, we use data from the Biobank, which is a large biomedical database in the UK. And we utilized about 30,000 subjects that we then split into females and males, because previous studies have indicated differences in aging between female and males based on biological sex. And then we did the normal thing of splitting them into training, the validation, and test sets. And we used that to train a deep 3D convolutional neural network. Now, we try to keep this as simple as possible. So the way the network was trained was to predict a subject's chronological age using only the 3D volumetric information from a contrast. At inference, this actually allows us to get an idea of the predicted brain age or the apparent age of the brain. And then we can use this further to get an idea of the brain age delta as the difference between the predicted and the chronological brain ages. What this tells us is that we either see acceleration in a person's age compared to the population norm if the delta is positive, or we see resilience to aging if the delta is negative. Now, all of this is good and fine, but where do we use this? Well, at first we went and asked the question, are all of these 57 contrasts available to us in the biobank capable of predicting brain age? And the answer to that is, well, yes, they are capable of predicting brain age. And we know that because all of these different dots representing different modalities fall under the red dotted line, which is the mean age failure case in which the network just predicts the average age of the population. So it's not capable of learning anything. But then what we see is that all of these different contrasts, 57, fall neatly into categories informed by their overarching modality. So that tells us, it's a sanity check. They all encode similar type of information, but not identical, because you see there are variations between the different modality groups, but there are also variations between the different contrasts in those modality groups. And this just tells us the mean absolute error, so how well the network predicts in terms of years. But if we select a number of contrasts, a couple from each category, these differences are also visible when we look at the distributions of their deltas. So this gets us thinking, okay, they have different accuracies at predicting brain age, but do they also encode different information about the aging brain, about the factors impacting the aging brain? So what we did to find out that was to go to another database in the biobank, which consists of biomedical and physical measurements and environmental measurements for subjects. And we correlated the brain age deltas that we've calculated for age contrast with these phenotypes. And we got these types of Manhattan plots. Now, in these plots, we have 17,000 different correlations with different biomedical and phenotypes. We grouped them based on their overarching category for easier visualization, and we plotted their significance, their statistical significance, against two metrics, the false discovery rate and the Bonferroni, as tests of significance. And this tells us, okay, there's something in there that might be interesting. But is this meaningful in any way? Doesn't this make sense from a biological point of view? Well, let's check this out. So for female subjects, we see very strong significances for the skeletal measurements. These are essentially bone mineral density measurements in the body at different locations. So let's just take one, bone mineral density. We see negative correlations between brain age deltas and the bone mineral density. So the higher the bone mineral density, the younger the brain appears. If we look at literature, though, for female subjects, we generally see that estrogen production premenopause is associated with higher bone mineral density, lower inflammation in the body, and younger appearing brains. At the same time, a loss in bone mineral density as female subjects age are also correlated with older appearing brains. Now, the linchpin in this whole equation is the inflammation, which has been found by numerous studies to actually correlate. So more inflammation correlates with older appearing brains. So looking at that entire equation, it makes sense that we're seeing those negative correlations with skeletal measurements. As another test of insanity, let's say, we also had a look at how the female group compares to the male subject group for this particular structural MRI contrast. And all of a sudden, we see that the skeletal measurements in pink aren't present anymore in the male subject group as significant associations. And that obviously is because we don't have that estrogen production component in the male subject. So this is actually why we've also separated the female and the male group. And this is all great up until this point, but now we ask ourselves the question again, are all of these contrasts different? So we had a look at how the number of significant hits varies for each of these categories. And here we have our previous data that we saw in the Manhattan plot for the female subjects, those in gray. That's our bone mineral density from before. But we see that in a lot of categories, we're actually not having any significant hits. Now, this is what happens if we populate that graph with information from some other contrasts derived from other modalities. And actually what we see there is that in all of these categories, so alcohol, blood assays, cardiac measurements, physical measurements, and we add in diet and tobacco for the male subjects, which aren't captured by this plot, we wouldn't have seen any associations in these categories of phenotypes if we were just sticking to the structural MRI data that all of the previous literature has been focusing on. And in fact, overall, we have found that we have 191 significant associations throughout our data set, and 140 of these are novel. So they come from contrasts and modalities that haven't been used before in literature. And we're going to be making all of this data freely available in the next couple of weeks. Just to touch on some interesting associations that we've been seeing, here, black means that that particular association is correlated with younger appearing brains, and red is associated with older appearing brains. Now, if we have a look here, so we have drinking frequency that shows up in functional MRI Makes sense. We know that drinking alcohol does impact the cardiovascular system, and resting state functional does look at information from the cardiovascular system. But then we have two interesting ones, the C-reactive protein and the cyclazine. Now, C-reactive protein is a measure of inflammation in the body. And we know that inflammation should lead to older appearing brains. But here, it's in black, so it leads to younger appearing brains. So we might ask ourselves, okay, what's going on here? And this is actually a cautionary tale of why none of the correlations that we're seeing should be treated as causation. Because in this case, it might be a situation where the subjects are taking anti-inflammation medication, and that actually is what we're seeing here. Cyclazine is also an interesting one because it's an anti-sickness medication, and as far as we can tell, there is no direct link to any biological process that should impact brain aging. So this could be a situation in which we see an off-target effect for a particular drug. Okay, so we saw all of this, but the next thing we asked ourselves is, okay, good and great, nice results, but can we get them even better? Can we combine these modalities in such a way that they use information from all of them or groups of them to get what we need? So we went back to our big collection of contrasts, and for all 57 of them, we've combined them, we've ensembled them at the subject level. And we did this in a few ways. We did naive averaging, where we just average predictions at the subject level. We did a few linear methods, of which the elastic net actually produced the state-of-the-art results. And we also looked at nonlinear ensembling with an MLP. We got state-of-the-art results in terms of accuracy, but what do you know? When we look at associations again, stuff starts missing in some of our categories. Now, we hypothesized that this could be because certain contrasts have opposing sensitivities to a particular phenotype of interest. So when you ensemble, you want to get the results as accurate as possible, but those opposing sensitivities are seen by the ensembling algorithm as noise, and it's eliminated. But we can also use that to our advantage. So if opposing sensitivities lead to signal elimination, similar sensitivities should lead to signal amplification. So we looked at the alcohol category, and what we did was select a few contrasts that all correlate highly with frequency of drinking alcohol, and we ensembled them with elastic net. And lo and behold, what you see, the elastic net ensemble in magenta over there actually produces a better correlation than all of its components combined. So we're managing to amplify the signal of interest here. And we've repeated this for a few other phenotypes, and we got the similar results. Now, all of these results were very interesting, and they got us thinking even more and more and more how we can use this information. Now, these are results that haven't yet been published, so this is a preview. But other stuff that we've been playing with includes deep modality fusion, where instead of training one network with one modality, we've trained one network with multiple modalities, allowing it to take advantage of complementary information. And the other thing is we went into Swin transformer architectures for prediction, because we realized that this architecture can pick up on different features than a convnet does, but also gives us inherent explainability by looking at the attention activation maps. So which parts of the brain contribute to a brain age prediction most? So to wrap up our journey, today we've seen that different MRI contrasts are capable of predicting brain age and encode different information about the aging brain. We found a lot of statistical significant associations that we're going to be making available to the public, and that hopefully will be informing causal studies in the future. And we see that ensembling brain age predictions lead to both better accuracy and the amplification of signals of interest. Now, you might ask, okay, Andrei, why have I been sitting here 20 minutes listening to you? Why does this matter? And it matters for three reasons. First one, as I said, our studies allow us to target causal studies later on based on our correlations to find actually what are the factors actually impacting brain age. This can be translated into the medical field, where we can use this for the treatment of neurological conditions. We can use this for targeted intervention for at-risk populations. But we can also be using this for monitoring off-target effects of different drugs, as you have seen from the cyclizine example from earlier. And the hope is that in the future, so medium term and long term, this will also translate into the political spectrum. That could be in the designing of better environments for aging populations, because we know aging is one of the major challenges of the 21st century. It can translate into prevention strategies for neurological conditions, and overall in designing better environments and cities for humans to live in as they age. So I'd like to thank you all for attending the talk today, but mostly firstly, I'd like to thank all of my wonderful supervisors from Oxford, from Roche, the wider teams, and my sponsors. And again, thank you all for attending, and thank you for your attention, and I'm open to questions now.
Do we have any questions? Yep.
Yeah?
Do you know why structural MRIs use that often compared to the others? Is it more cost intensive, or is it
It's just more available. So structural MRI scans usually take around 3 to 5 minutes to acquire. So the majority of the databases in the world just acquire structural MRI scans and maybe 1 or 2 other modalities. But they never try to have a very rich database of modalities. The UK Biobank, because it affords to take a bit more time with the subject and they accept to be scanned for about half an hour, is able to collect all of these different modalities, which are then processed into all the contrasts available. I think, yeah.
I don't know the data from Biobank, but I was wondering whether they have longitudinal data and whether these models could at some point be predictive for the aging of somebody that's young and how it's going to look.
That's actually a very good question. So the Biobank has had funding a couple of years ago to start acquiring a second time point scan 2 years later for about 4,000 subjects out of the original 40,000 that were in the imaging database. And recently there has been funding approved for another time point, another 2 years from now. So we're starting to accumulate longitudinal data for that. So that is something that is happening, yes.
Please.
So thank you very much for your talk. I have 2 questions to the data. One is how distributed is the ages from your data, and also were these all healthy subjects or?
Yes, so the data is almost normally distributed between 40 years and 80 years old. I'm saying almost normal. The Biobank tries to follow the distribution of the ages for the UK population to get a significant view of the UK population. When we sampled the data, we were very careful to match the underlying distribution of the population so we don't end up with under or overrepresented classes. Sorry, what was the second part of your question?
Were these all healthy participants? Or?
So they are self-reported healthy, but they do go through a medical exam. So for example, there might be subjects with diabetes in the Biobank, which has been diagnosed by a doctor, as an example.
Thank you. We have a question here at the back with a colored shirt. Yeah, sorry to.
Thank you very much for the very interesting talk.
You're welcome.
Information fusion, could you identify the contribution of different modalities in the final prediction? And one more question, since I'm not an expert in MRI, is there more radiation or like is the correlation between the enhancers of predictions when you use more modalities compared to just the structural MRI?
Right, so let's go first to your first question. So for the fusion work, we are exploring different fusion strategies. Now, if you do very early fusion in a deep learning net, then it's a bit harder to get an idea of the relative contribution of the modalities, but you can get an idea by looking at the filters of which features are contributing in with methods like CAM. It's still a very open research question, but we are seeing different results when we ensemble them with deep fusion than when we're looking at individual ones. So there's probably some information transfer between them. Regarding the MRI question, there is no radiation in there, it's just magnets. I'm not an MRI expert, I'm the deep learning guy in the team. But my understanding is you essentially modulate the power and the frequency of the magnet and of the radio waves that you use to excite water molecules in the brain. And that gives you different information in the images. So you can look at the oxygen, or you can look at the water and the diffusion, or you can look at the structure of the brain.
Okay, last question.
Yeah, thank you very much for the talk.
You're welcome.
If you don't have the chance to have all modalities,
Mm-hmm.
which is I think usual in the practical field, can you say what would be, or can you identify which ensemble would make the most sense to basically predict brain evolution, so that it is more feasible in like clinical work?
That's actually a very good question. At first, as you saw, each of these single contrasts is able to predict brain age in its own sense. In regards to which ensemble to use, it's a very open-ended question because it depends a lot on what you're trying to look for. So if you're trying to look for the highest accuracy, so the closest you can get where you're only left with highly structural biological errors, the ensemble that worked best for us was one that used all of the modality, all of the contrasts, minus the ones coming from resting state. But if you're looking at getting particular information about, for example, alcohol consumption, as I showed earlier, in alcohol consumption, there was a very specific group of contrasts which all correlated with alcohol. So an advice would be, if you're interested in seeing the effects of alcohol on the brain, just use those particular contrasts. The biobank is accessible to all researchers upon application, so it is open in that sense. So people can access the data to do further studies on that.
Okay, I would like to thank Andrei again for this excellent talk. Thank you
thank you,
very
everyone.
much. I give you this
Oh, thank you so much.
purple, and I think now we have a